Welcome. Good afternoon, everybody. Thanks for joining us. My name is Paul Anderson. I'm the Executive Director for the Maine Center for Coastal Fisheries. My pleasure to welcome you to today's Lunch and Learn webinar. We do these monthly here at MCCF on various topics involving our programs and our partners and stakeholders. Our mission, many of you probably know, is to help sustain commercial fishing in the communities in Eastern Maine that depend on fishing. As you all know, there's a confluence of huge issues facing our coasts and our coastal communities and the fishing industry that we cherish and really depend on in our work to try to support them. Is done in a number of ways, all of it collaborative. We do collaborative research work, collaborative management work, and collaborative education. And today we're going to focus on one of the projects that fit into our collaborative research work. And Pat Shepard is our collaborative research specialist. So he's on point here today, and he and with a little help from Tate Yoder are going to help present some of the fascinating work that we're doing there in that space. Couple quick updates for those of you in the area, in the community. We are pleased to be opening our Discovery Wharf Interpretive Center tomorrow, actually, for the season. And if you've been there before, you know it's a great little facility set up with a touch tank for families to enjoy. But we also have a virtual reality set up and a touch wall and a number of other things that you can see and experience to learn about commercial fishing in Maine. We have a brand new set of interpretive signs out on our walkway on the water side of the building that just got put up. In fact, Pat oversaw the creation of those. It's 4 signs talking about some of the features of fishery and Stonington as a fishing community. So if you're down in the area, please check that out. We are opening the Discovery Wharf 4 days a week by reservation, preferably on Tuesdays, Thursdays, Fridays, and Saturdays. And they will feature, of course, Captain Leroy Weed, who will be sharing his knowledge and stories and his charm with visitors. So if you haven't seen the Discovery Wharf or met Leroy, think about that for your summer trips to Stonington. We are recording today's session, so if you want to see it again or you want to share this experience with your colleagues and friends and family, you'll be able to do that within a week or so. We'll have it posted out at our website with all of the other previous Lunch and Learns. And we'll do this again next month at the end of June. June 24th would be the next Lunch and Learn session. So thanks for joining us. I'm going to hand it over to Pat Shepard now, and I'll be in the background and we'll help with Q&A at the end of the hour. Pat?
Thank you, Paul. I am Pat Shepard. For those of you who don't know me, I'm the Collaborative Research Specialist here at Maine Center for Coastal Fisheries. I'm actually from here in Stonington. I grew up about a half a mile from here in a fishing family, and spent the whole first part of my life lobstering with my family, and ended up leaving home, going to the University of Rhode Island, obtained a bachelor's degree in marine affairs down there, and ended up eventually working my way back home and getting a job about a half a mile from the house I grew up in. So feel pretty fortunate to be here in Stonington, and I've been here at Maine Center for Coastal Fisheries for about the past 10 years, this past April. So if you've heard me talk before, especially in Lunch and Learns, you've heard me talk about the Sentinel survey that we run for groundfish in Eastern Maine. And if you've heard that talk before, you're in for a treat, because we're going to talk about it again. Today's talk is going to be a little bit different than the ones that I've done before. This is going to be about the faces of collaborative research and the different people that participate in collaborative research, in particular on the Sentinel survey that we run. And the first thing that you'll notice in this first slide, I kind of put together a collage of a lot of the different people. This is kind of only scratching the surface, but a lot of the different people that work on the Sentinel survey collecting groundfish for our Sentinel project. And the first thing that you'll notice is these people are having a terrible time. It's awful, grueling work. None of them are happy. None of them are enjoying their work. No, I'm just kidding. They, if you can see their faces, they're all really excited to be here. Actually, the upper, I don't know where it is on your screen, but the upper left-hand side It's actually a father and son team, and that's actually taken from the same photo. They're both screaming about a halibut that they caught that day, and I've got a series of photos that I'll show at the end of the presentation and tell you a little bit of tell you a story about that. So the any collaborative research project starts with a curious person, somebody who is just really curious about something and asking a question about something. And the Sentinel survey started with a man by the name of Ted Ames. And he also happens to be the founder of Maine Center for Coastal Fisheries. Here he is on his lobster boat that he had a few years ago. He's been a draggerman for groundfish. He's gone shrimping. He's pretty much done every fishery under the sun. But what really got him curious in the later part of his career and still keeps him curious today is the mechanisms for recovery of groundfish in eastern Maine. And about 10 years ago, he got together with a man by the name of Jason Joyce down in Swans Island. And as I'm doing this, I'm going to be starting to paste these faces up on the wall behind me. And hopefully by the end of this, we'll see this sort of start to fill up with people, because that's kind of the point of this, is showing all of the different people that work on this project. So Jason Joyce and Ted Ames got together and started talking about how they would go about going and collecting groundfish for research purposes. And Ted had a lot of knowledge about ground fishing from his days, and Jason had a lot of knowledge from his time out working on boats. And they just started to kick around a project where they could go out and sample these things in a way that would collect data over the course of time. And they needed a regulatory partner. It wasn't John Hare at the beginning, but it is now. But the National Oceanic and Atmospheric Administration partnered up with us very early on. And what happens when you take fishermen and scientists and couple them with regulatory partners, what happens is you can identify research priorities. They help you identify research priorities. Oftentimes they can help provide funding for the project, which is what NOAA did early on. Help strategize goals and objectives that not only help the fishery, but help NOAA with their work as well. And it really grounds the research in realistic management opportunities. So when we start to talk about goals and objectives for any research project, usually there's a management objective at the end of it. And partnering with regulatory partners early on can sort of help steer things in that direction. So keep going here. Here's Jason Joyce. He's out of Swans Island right there. And John Hare is down in Rhode Island. So we're going to start filling this map up with faces. Another partner earlier on in the onset of this project were Jeff Smith at the Nature Conservancy, and later on Jocelyn Ronnebaum joined, and they worked for the Nature Conservancy, which is a conservation organization down in Brunswick. And really what they've provided to the project is not only as a funding partner, but we meet with them over the course of the year, multiple times a year, and they provide advice to us. They provide insights on what they're learning from a different part of the coast. They also participate heavily in the New England Fishery Management Council, so they can offer us a connection to other aspects of the industry and insights from different areas. And we've been really lucky to have them as a partner. So here's Jeff. Here's Jocelyn, where's Brunswick? Right about there. Moving along. A very early partner and something that we realized we needed earlier on was a university partner. And we needed a university partner for this project for a bunch of different reasons. One, the credentials to collect and analyze scientific information. Really nobody on staff here at Maine Center for Coastal Fisheries had the expertise to do that. But the other major part of it is Maine Center for Coastal Fisheries is what's considered a boundary organization, and we work sort of in the spaces between science and management and government, and we're not defined in the law as a scientific institution. So what a lot of people don't realize is we can't actually collect and analyze scientific information ourselves. We have to partner with a university, with a scientific institution, to be able to hold the permits to get this work done. So the other thing that the university provides is the ability to publish findings, provide graduate students to the project, and access university resources, which has been majorly helpful in this work. So we've got an interesting mix of people here at the onset of the project. We've got stock assessment scientists at the University of Maine. We have fishermen. We have sort of a curious fisherman turned scientist at the beginning of the project. We've got funding partners from the Nature Conservancy. And so we've got a really interesting mix here at the beginning of people from different walks of the industry and people from different corners of the industry. And what you can imagine is when you get different people like that in a room, it kind of starts to turn into a Tower of Babel situation where, God comes down and he punishes everybody by speaking a different language. And what projects like this really need, collaborative projects like this with many different stakeholders, is a coordinator. Somebody who can build relationships with partners, somebody who can be in the room to help translate what the other is talking about, so that when we're in our planning meetings and fishermen are saying things like 12-aught mustad hooks and rollers and scrubbers, you've got to have somebody in the room who can lean over to the stock assessment scientist and explain that kind of stuff. At the same time, when stock assessment scientists are talking about models, you can, at least for me, I can lean over to the fisherman and say, don't worry, I don't know what he's talking about either. Trust me, he knows what he's doing, because I don't know a whole lot about that stuff either. But really a person who can sort of keep the wheels on the bus, facilitate trips. A lot of times the coordinator or the boundary organization can act as the fiscal agent. It can recruit partners and participants, and really create that synergy that makes it all kind of tick. So moving along here, there's Walton University, and that's in Portland. And here I am in Stonington. What do we got next? I don't work alone in this. I have a lot of people here on staff that help me out. And that's sort of what it's all about. Our director, who you just heard speak, Paul Anderson, he has the fun job of fundraising for this important effort, and connecting with funding partners and getting them excited about the work and coming and experiencing it. Dr. Carla Gunther, our chief scientist here at MCCF, she is really good and really responsible for sort of the higher level scientific questions that we're asking in this project, and connecting with those different scientific partners to recruit them to the program. Mike Thalhauser helps out here and there, and gives us a connection to the freshwater habitat, which I'm going to talk about in a little bit. And our education coordinator there on the right, Tom Dime, really what he provides to the project is usually some good entertainment and ability to catch some fish on the boat. So we've got it's really a team effort here at MCCF, and even though I'm sort of point guard on it, there's a lot of different people just here in-house who help out with that stuff. So we're going to step down right around here. So that's it, right? That's all we need for people. Just kidding. There's a lot more people involved. So now that we have sort of a group of people who can coordinate the effort, here's what we've been doing for the past 10 years. This is an example of really any particular year on the survey, what you're looking at is a map of the stations that we visit on any given year. So the red sites that you see sort of in closer to shore are jig sites. We steam out to those sites and fish with rod and reel for groundfish. And the blue and yellow sites that you see offshore are, we use a combination of jig and longline to fish those sites. Since the project began, when the project began, we really only used one or two fishermen to get this done. And since then, we've added a lot of fishermen to the program. So my helpful assistant, Tate, is going to help me put some fishermen up on the board here. And you're going to start to see this map sort of fill up with people. And these are all people, you know, from different walks of the industry, from different corners with different interests, but they're all heavily invested in the question at hand, which is how do we learn about cod in a way to help them recover in Eastern Maine, and how do we sort of tease apart what we need to know to be able to facilitate that happening. And so for the past 10 years, we've been steaming out to locations just like these, deploying gear, catching fish. And I've got a short video here to show you about what we do on board.
Cod, and I take a whole bunch of measurements and samples from this one, since this is one of our target fish on the Sentinel survey. So first we're going to measure it, and this one is 46 centimeters. Put that right down on my data sheet. So we're going to weigh it. This one is 2 pounds. I'm going to take a photo of this, which we call a morphometric photo. A researcher can look at the shape of its body, and so kind of like a combination of like the length and the width and how far different body parts are from each other. And they've actually found that that can be different based on where the cod lives. The next thing that we need to do is figure out the sex and maturity of this fish. And this one is a female. You can tell right here. These two things are her ovaries, but she's a juvenile, so she isn't actually developing eggs yet. So they're like very small and very pink. So she's a juvenile. Now we're gonna take the stomach out. Here's the stomach. It's hard to tell. There's probably something in there. This is 95% ethanol. So basically this will preserve that stomach until I'm ready to cut it open and analyze it. And a lot of times I have to use a microscope because they eat things like little shrimp, and I try to identify them as much as I can. But other things we find in the stomach, they eat a lot of crab, especially like Jonah crab, or we find lobster. Sometimes we'll find little sea stars. It's pretty cool to see what they eat, and the larger they get, the more likely they are to actually find little fish, like whole fish, in their stomach. So we'll see when I end up analyzing that one. Two other samples that we end up taking are otoliths, which are ear bones within essentially the fish's head. They're going to come out of like right here. So what I'm going to do is actually just cut off the whole head. And then later, once the survey is over, when we're on land and in a lab, we'll extract those. But the really interesting thing about their otoliths is that we can actually figure out how old a fish is by looking at their otoliths. if you cut them, they have these rings, kind of like a tree, and you can count the rings and see how old they are. We'll also actually take a muscle tissue sample from like right here. It's kind of similar to looking at the stomachs in that it can tell you what they've been eating, but it tells you what they've been eating over a longer period of time. So the last sample that we take is one of these fin rays from their caudal fin, their tail fin, kind of like, you know, how hair or spit or whatever from a human gets you DNA information. This is where we get a lot of the DNA from fish, is from these fin rays here. So I'm just gonna cut that one off. This is also going in ethanol. We're really interested in where they're reproducing, so like where they're coming from. And we're not really sure where the fish in the Gulf of Maine, in the eastern Gulf of Maine, are coming from, because we don't really think that too many of them are spawning around here. But by looking at this DNA evidence, we can actually kind of tell where they're from. And if we were able to figure out that they are spawning in the eastern Gulf of Maine, that would be a huge deal, and really good news for cod in this area, because we think that most of them spawn in like the southwestern portion of the Gulf of Maine. There's a lot of really awesome information that we get from taking all of these samples. And these different samples go to a lot of different researchers all over New England.
So that's a quick rundown of basically what happens every time we go and visit one of these sites. And like I said, we've been doing this for about the past 10 years. And the slide that you're looking at right now is basically everywhere where we've fished over the course of the last 10 years. And the circles that are colored in red are all the places where we've actually found cod. And one of the things that you'll notice is that that green line that sort of skirts the coast is the 50 fathom edge. And we've caught the vast majority of the codfish that we've caught on the survey inside that line. So shoaler than 50 fathoms or shoaler than that contour. So we've been doing this for, like I said, the past 10 years. And now I'm going to get into some of the different things that we've learned. Robin mentioned in the video that all the samples that we take, we ship to basically the far corners of New England, and different researchers get a hold of these samples and they analyze them for different things. One of the easiest things for us to analyze is length frequencies. And we do this pretty much right here in-house. We have all the data that comes in off of the boats, and it's really just a matter of plotting it on a graph. And so what you're looking at here is all the different length frequencies of the fish that we've caught over the past since about 2010. Cod reach reproductive maturity, or we think cod reach reproductive maturity right around 60 centimeters. And almost all of the fish that we've caught over the past 10 years have fallen below that line, with the exception of 2014. We caught a few that we would consider to be adult-sized fish or mature fish. Most of the fish that we catch are right in the size range of the fish that Robin's holding up over on the right. The stomach samples that we've taken, Robin has analyzed some of them. Some of them have been analyzed by other people at the University of Maine. Robin Linear she started her career at the University of Maine and has transferred to Stony Brook, but she remains involved in the project. We're lucky to have her on board and still on board. She last year analyzed about 200 cod stomachs. We had 200 cod stomachs. She analyzed about 93 of those. And this is the collection of prey that we found in the stomachs of the fish. So about 60% were crustaceans. And this is exactly what we would expect from a juvenile population of fish. They're eating things like Jonah crabs and rock crabs, different shrimp, lobsters, things like that. Not a whole lot of them were eating fish. Some of them were actually found with rocks in their bellies, and this is one of the cool stories about collaborative research that sort of gives us an example of what we can learn from fishermen. I mentioned to one of the fishermen that works on the project that we found rocks in the bellies of some of the codfish. And he said, you know what? I heard an old timer talk about the fact that just before a big storm or a big blow, codfish, halibut, and some other species of groundfish will actually eat rocks to keep themselves ballast and hold close to the bottom in turbulent water. And I was like, you've got to be crazy. I was like, they probably ate the rocks by mistake. And so I got curious about it, and I went back and looked at the data, and it's actually true. The rocks that we found in the bellies of the codfish, I looked at the weather on either side of those capture events, and on either just before or just after we found rocks in the stomachs, the wind blew just about over 20 knots. So maybe it's true, maybe it's not. Maybe they eat rocks for ballast. Maybe they made a mistake, but it lines up with the data, and that's the fun thing that we can learn by working so closely with fishermen. Is we can take some of this data back to them and say, "Hey, what do you think of this?" And they can offer us their insights from basically an entire lifetime of being on the ocean. So here's another example of some of the stomach contents that we found in the bellies of cod. Like I said, most of them were crabs and lobsters. There were 23 different fish present in the 93 samples, and the fish that we did find in the bellies were Acadian redfish and cunner. We thought we found some herring, and that's actually what we've been looking for all along. Is we've been looking for these codfish sort of preying on herring, because we know that herring is, and in particular river herring, is an important historical prey base for cod. So that's one of the things that we've been sort of paying attention to and looking for in the bellies of cod. And I'll get into that a little bit more in the next couple of slides. One of the other partners that we have, lacking on my posting here. This is, actually, I think her office is in Booth Bay. This is Becca Peters. She heads up the Maine, New Hampshire Inshore Trawl Survey. She's the sea sample that goes out on all of those trips. She collects cod stomachs from hake and cod and halibut. And this year we're going to be working with her to get her some of our stomach contents as well. Her project is primarily to determine the potential predators of juvenile lobsters. So she's looking for, in particular, she's looking for lobsters in the bellies of cod and halibut and all those other species. But she obviously analyzes everything that comes up in those samples. So this is a new partnership, and somebody else that we've added to our wall of people here on the survey. Greg Puncher up at the Department of Fisheries and Oceans in Canada has taken a lot of our genetic samples, our fin clip samples that you saw in the video. And a few years ago, he analyzed about 248 adult and 215 juvenile cod, and actually found that there's a genetic distinction between the cod that we see here in the eastern Gulf of Maine and up in the Bay of Fundy that are genetically distinct from the rest of the Gulf of Maine. And that's important because it has implications for cod management and the actual stock structure of cod in the Gulf of Maine. Similarly, Adrian Kovach from the University of New Hampshire has analyzed some of our genetic samples. Basically, we take two different sets of fin clips from the tail of the cod. Half of them go to the DFO in Canada and half of them go to UNH. And Adrian Kovach found basically the same thing, that cod in the eastern Gulf of Maine they have a genetic distinction from the rest of the New England region. So when management conversations happen about stock structure for cod, this is the kind of information that can help steer those conversations. Our partner down at the Woods Hole Oceanographic Institute, Simon Thorold, has analyzed a lot of our muscle tissue samples from cod, haddock, pollock, mackerel, and halibut. And what you're looking at here is what's called a constellation plot of the stable isotopes that exist in the muscle tissue. So basically, Simon can take a look at the muscle tissue that comes from just in front of the first dorsal fin, and he can analyze the chemicals, the stable isotopes of carbon and nitrogen, and figure out basically diet patterns over the recent history of that fish's life. And so I don't know if this is hard for you to see on your screen, but you can basically see different groupings of species as you move around this constellation plot. And you'll see that cod are sort of grouped with mackerel over on the left there. And then you've got white hake that are sort of kind of on their own. You've got a group of haddock, pollock, and mackerel sort of in a cluster, and then pollock on their own. And then as you move around this, you can see sort of clusters of species that are either feeding on things independently of other species, or they're grouped together with other species, meaning that they're feeding on the same types of things. Our purpose for analyzing these muscle tissues was to figure out if any of our cod or any of our groundfish in general are feeding on alewives or river herring. We're trying to figure out if all of the dams coming down on the rivers is actually having an impact on predators in the bay. And I don't know if you can see it. I can't see it on my screen, but there's a line that sort of goes straight up, and there's a river herring up at the top of that constellation plot. Basically what that means is we haven't found any alewife or any freshwater presence in the muscle tissues of any of our fish. But when you sort of couple that information with the other information that we've collected and the other information that's been analyzed by some of the other partners, you can sort of piece together reasoning why that might be. So we're looking for fish in the bellies of these groundfish, but what we've sort of determined is that the fish that we catch on the Sentinel survey are too small to be able to accommodate fish in their diets. The predominance of juvenile fish on this survey sort of preclude them from having a piscivorous diet or a fish diet. So that's sort of led us to, again, go back to our fishermen partners and present this information to them and say, what do you make of this? We're catching a large number of juvenile fish. We can't seem to find adults in the population, and because of that, we're not seeing what we would expect to see, that being alewife presence in the bellies of these fish. And what our fishermen partners have sort of said across the board is that maybe we're sampling at the wrong time of year. They obviously fish these waters year round. They are paying attention to the comings and goings of different species in the area. And they've told us that the adult fish, the adult cod at least, will come in and spawn during the winter. Our survey only runs from June through October. So it's not that we don't have adult codfish here in the Eastern Gulf of Maine. It could very well be that we've just completely missed them because of the timing of our survey. So one of the neat things about collaborative research projects like this is you can take that information to the different corners and the different partners and say, okay, how do we change this and how do we accommodate it moving forward? And what we've been able to do is extend the survey into the shoulder seasons. And this year we're actually going to be sampling, whoops, I went too far. We're actually going to be sampling in the wintertime to try to catch those adult fish to see if the rest of our hypothesis about alewives holds true. One of the other partnerships that we've been able to gain over the past, let's get Greg on here, up in Canada, Adrian down at UNH, and Simon at Woods Hole. One of the other partners that we've been able to gain out of this work is a lady by the name of Michelle Staudinger. And Michelle, if you're on here, I apologize for botching your last name. She's collecting marine bird shell samples from the Penobscot Bay region. And we figured if we couldn't find what we were looking for in fish, we would partner with somebody who is looking for similar things in other species, namely birds. So if groundfish are unable to benefit from the rapidly recovering river herring resurgence, maybe other important species are, like seabirds. So that project has just started up as well. As I said a few moments ago, we've been able to sort of take this collective of information back to our partners and sort of make small redesigns in the way that we sample fish, so that we can be the most efficient. We can collect the information that we need to collect without spending a ton of money doing it. And what we've learned is that if we catch most of our fish inshore, we're going to focus on the inshore environment, at least until things seem to be looking up in the offshore environment, and then we're going to move back offshore. But we're really going to focus for this next few years on the inshore environment, sampling into the shoulder seasons to try to capture those older fish, and hopefully we can prove that dam restoration, river restoration, and dam removal is having an effect on marine predators in the bay. And with that, I'll open it up to some questions. And if somebody can facilitate the questions, I haven't seen any come in the chat.
Thanks, Pat. Thank you. Nicely done. Folks, you're welcome to raise your hand or enter a question in the chat or the Q&A. And we don't have any right now. I enjoyed that, Pat. Pat's always very creative about how he presents stuff and writes about the work that we do. And I thought that was good. And we taught a young 24-year-old who Vanna White is. Kind of. I don't even remember the program that's involved. Is it like Jeopardy or something?
Wheel of Fortune, I think.
A Wheel of Fortune, right?
Okay, if we don't have any questions, I'm going to share sort of a favorite story of mine from the Sentinel survey. We still got participants here, so I'm going to share my screen again. So one of the most rewarding parts for me is getting to see some of our fishermen partners actually take their kids out on the boat with them and teach them sort of a new way of fishing or a new catch. Let's see if I can get there. I've lost my slide controls again. Maybe I won't. Oh, here we go. So this is Captain Matt Trundy, and he's out on his boat, the fishing vessel Savannah Says, and that's his boy standing next to him there, Wes Trundy. And you can see the big bend in the rod there. This is about 6 o'clock in the morning. We were about 20 miles south of Stonington, and this is probably our second drop of the morning. And Wes had the rod and reel in his hand, and he hooked up on something really heavy on bottom. And he starts hollering to his dad, "Dad, I got a fish, I got a fish." And you know, his dad, you know, just sort of poking fun at him. He's like, "No, you don't have a fish. You must be hung up on something on bottom." And so he grabbed the rod out of Wes's hand, and the fish took off and the rod bent over. And they fought this fish for about a half hour as it's coming to the surface. And they kept handing the rod back and forth, and Wes would get tired and he'd hand it to his dad, and then his dad would get tired and hand it back to Wes. And they finally got this fish up to the surface. And I don't know if you can see it in the water there, but there's this beautiful halibut on the bottom of the jig. And we have never caught a halibut on rod and reel before, not on the Sentinel survey anyway. And so this was a pretty exciting moment for us. You can see Wes there, the big grin on his face, just sort of anticipating that fish coming up. we happened to have Tate Yoder on board that day, and he took a series of photos that really kind of captures the moment. And so here's the fish coming up toward the boat. And then the fish being landed in the boat. And if you zoom in on Matt's face and on Wes's face, you can just see the excitement, and you can kind of feel how awesome it was to be on board with all the hooting and hollering when that fish hit the deck of the boat. There's the next one in the sequence. This fish was, I believe it was around 45 inches. So it was just over legal size. The legal size for halibut on the coast of Maine is 41 inches. So it just made the mark there. A little high five between father and son, probably one of my favorite pictures in the world. And quick measure to make sure it's legal. And a boy and his fish. Doesn't get much better than that.
Nice, Pat. Great. We have a few questions that have come in, and I'll try to get through these. One of our attendees had several questions. One, are there any tools, devices, or sensors you wish you had or have identified but are out of reach because they're either too expensive or too difficult to use?
Oh, that's a good question. We've talked before about acoustic sensors actually for sensing codfish in the water. If we could actually sense cod in the water. See, one of the problems with this platform is we don't have enough spatial coverage and we don't have enough temporal coverage to be able to go out and fish everywhere all the time. So if we had sensors in the water that could actually detect codfish or codfish songs, codfish actually do make noise when they mate and when they're in the near shore environment. If we had sensors like that to actually passively detect them, it would be pretty awesome. But we haven't developed the thinking around that and what it would take to put a bunch of sensors out there. I'm also wondering if there's some eDNA work that we can do on some of the spawning grounds for codfish. I don't know how well eDNA holds up in a marine environment. We do it in the freshwater environment, looking at the mud and silt and stuff on the bottom of lakes. And I've kind of wondered to myself whether we could do that in the marine environment in a discrete spawning ground to see if there's presence of cod where we haven't sampled before or don't have the means to sample. So.
Okay, that's a good idea. The eDNA group at UMaine is exploring those very questions. Robert Ferraro is asking about what about juvenile alewives? Wouldn't immature cod be big enough to eat them, or do juvenile alewives stay in freshwater away from the cod?
That's a good question. So what we've learned from the founder of the organization and the founder of the project, Ted Ains, is that cod typically feed on crustaceans when they're young because they're a lot more available. They're close to bottom. They don't have to chase after them a whole lot. They can sustain themselves on a predominantly crustacean diet for the first part of their lives. But as they reach reproductive maturity, when they get to that 60 centimeter mark, they actually need a fatty, lipid-rich prey to be able to spawn. They need the energy from that fat to be able to do their thing. And so it's got a lot to do with the size of the fish and the availability of prey. So if they're not of a certain size and the prey isn't available, they're either going to die or they're going to go somewhere else, which is what we're trying to figure out through the genetics work and the morphometrics work and things like that.
Right. So that's also kind of a fish behavior and bioenergetics question too, because they want to expend energy catching moving prey when they have to, like you say. But when they don't have to, maybe they want to eat sedentary prey like crustaceans and stuff. Hello to Jim Saltonstall. Nice to see you here. Welcome. I don't know if you're back up in Maine yet, but hope to see you. Jim Manning is asking, Pat, do you take any temperature observations during the surveys? If not, would you like to try some new sensors we have coming in 2022?
Yes and yes. We do take temperatures. We use tidbit, onset tidbit probes, and we hang them on the anchors of all the trawls, and we drop them down with the jigs that we drop. And so we do have temperature data, I believe, starting in 2013 or 14, we started collecting temperature data. But I'm definitely curious to learn about new sensors. And I was just about to order the new Bluetooth sensors from Onset because they're a lot easier to harvest the data off of them. But let's connect about what you've got in mind before I pull the trigger on that. Thanks for that.
That's great, Jim. Thanks for being here. Let's see, Zach Cliver has an interesting set of questions. He wants you to speculate about the return of cod to Down East Maine. I don't know if you mentioned Lisa Kerr's recent work. Do you think it's possible for the adult cod to return even with impending climate change? And do you agree with Ted that when and if a fishery does recover, we should prosecute it with hooks?
Yes, good question. I just based on what I've seen on the water over the past couple of years, we have seen a pretty marked resurgence of at least juvenile cod in the inshore habitat. We caught more cod last year. I think we caught more cod. I was looking at one of the days. We caught more cod on one station than we did in the previous entire year of the survey. So we are seeing a resurgence of juvenile cod. We just have unanswered questions as to where they came from. My feeling is that they were spawned there. They didn't move there from someplace else, but there were adults that came in and spawned on these shoal cobbly habitats over the course of the winter. And then the adults took off and left the eggs there because we haven't been able to return the adult fish. I forgot the second part of the question, Paul.
If a fishery were to return, do we agree with Ted and his notion that we ought to prosecute it only with hook and line?
My opinion on that is hook and line all the way. I think it produces a better quality fish, and this is not to say anything against draggers or gill netters. We need those segments of the fleet too, but in an environment where we don't have a whole lot of fish, and we don't have a whole lot of fish coming back at a very rapid pace, we need to be very careful about how we harvest that resource. And if we can pick and choose by harvesting live fish and releasing juvenile fish that are still alive, then we can select for those larger fish that we do want to harvest if and when a fishery comes back here. And it's going to be of the highest quality so that we don't have to land volume. We can land value and quality.
Okay, thanks. A couple of questions I'll combine back about the method on board the vessels. Are there particularly time-consuming processes during the survey that we wish we could automate? And also similarly, are there things we'd like to be measuring that we can't right now?
Yeah, there's so one of the reasons that we're focusing on the inshore environment is just that. There are a lot of time-consuming parts to being able to fish offshore with tub trawls, with long lines. If you drive by MCCF at any time during the summer, you'll usually see a tent out front and me standing under it baiting hooks. And I do that in between every single trip over the course of the summer. And it takes a long time to bait all those hooks and get them ready for the next trip. And that's one of the things that we decided to pare back on this year and focus on just jigging, because it takes a lot less effort, a lot less equipment, a lot less time to go and jig as it does to go and set baited tub trawls. And that's one of the things that we're focusing on this summer. Second part of that question, I already forgot.
Things we'd like to measure, but we can't right now.
That's a good question. I would have to think about that. Who asked it? We'll get back to
anonymous attendee.
Oh, okay.
Let's see. We have one about the lobster fishery, but let me first go to one from Trey. He's asking if lobstermen are reporting seeing cod in their traps.
That's a really good question. Lobster fishermen typically do catch fish, although the lobster traps aren't really designed to catch fish, but they do see fish come up in their traps often. And we do lean on that information as far as being able to tell sort of what's moving into an area and what's moving out. And I see there was another question about whether we use lobster fishermen in our work. Was that earlier on?
Well, there's one that I think is asking in general, are we working with the lobster industry?
Yeah.
And looking to coordinate large-scale fishery projects in general.
So in particular in the Sentinel survey, the project that I just got done describing, we do all of the fishermen, almost all of the fishermen that we use to collect information about groundfish are commercial lobster fishermen, and they do this project sort of either in their downtime or early in the spring when there isn't much going on in the lobster fishery, and we lean on them quite heavily and use their boats to do the work. All of the fishermen that you saw me paste up sort of on the wall here are all commercial lobster fishermen that do this project on the side, and the neat part about that is I've been able to sort of convince them to do it on a volunteer basis and spread the work out to 9 or 10 different fishermen. And they do it because they're curious too. They want to see what's going on with different resources in their area. It gives them access to a different gear type that they don't typically get to fish with. It gives them access to species that they don't typically get to catch in the course of their lobstering endeavor. And I think it's a lot of fun for them to sort of break the lobstering routine and go fish for something different. So.
Great. I think we made it through all of our questions in chat. Thank you, Pat, for a great presentation and your work. Thanks to Tate Yoder for making all the electrons go where they need to go. And thank you to all of our attendees. We appreciate your attendance and interest and your support. So thanks. Check us out on the line. Check us out in Stonington, and we'll be back with another Lunch and Learn on June 24th. I hope you enjoy the rest of your day.
Thanks, everyone. Appreciate it.